Today we're going to revise proteins. Now, as with all the other biological molecules, we need to know what monomers proteins are made of. Proteins are made of amino acids. And you need to know how to draw an amino acid. Okay, so the easiest way to remember the structure of an amino acid is that it rhymes. You've got NH2, CHR, Q. So that's NH2, CHR, Q. Now that's the generalised formula for amino acid because we've put an R on there. Now R isn't a chemical symbol, it stands for the random or the variable group, and that differs with each amino acid. The simplest amino acid just has a hydrogen there, and that's called glycine. So the R group of a hydrogen is the simplest amino acid called glycine. Okay. Now if we want to join amino acids together, we have to draw two of them out next to each other. So I'm going to draw another one next to this one. Okay, so again, I've got my NH2, CHR, Q. And hopefully you can straight away see with this condensation reaction where the water is going to be coming from. Okay, so here's my H2O. So water is one of the products, and that leaves me with two amino acids joined together, and that's called a dipeptide. So if I draw out the remaining dipeptide, I've got NH2, CHR. Now on this carboxylic acid group, we've lost the OH, so we've just got double bond to O, and then we've got a new bond formed to the nitrogen of the second amino acid. Only one hydrogen left there. CHR again, and the final Q. So in this condensation reaction, we've had the release of water, we formed a larger molecule from two smaller molecules, we formed a dipeptide, and we've got a new bond. This carbon-nitrogen bond here is called a peptide bond. Okay. Now the polypeptide of proteins are called, obviously, polypeptides or proteins. And all that happens is we have repeated condensation reactions. So a polypeptide has more than 3 amino acids joined together by peptide bonds. Okay. So with polypeptides, they fall into different categories of structure. So what we want to look at now are the different categories of structure. So proteins have 4 levels of structure. So the first one is primary. Then we've got secondary, tertiary and quaternary. And each of these has its own definition for you to learn. Okay, primary structure is assembled at the ribosome. You learned that in your cell structure unit. And the way that it's assembled is we've got amino acids which are joined together by peptide bonds. So the definition for primary structure is the sequence, type and number of amino acids in a polypeptide chain. And remember, the bonds that are important here, there's only one type of bond, and they're peptide bonds. Okay. Secondary structure takes that level of structure a little bit further. So we've already got the chain of amino acids. This time we have simple folding of that chain, and we fold into alpha helix, which is a coil, or beta pleated sheet, which, as its name suggests, is a pleat. Now, if they were just originally chains, we don't want them to fall apart again. So the way that they're stabilised is if we draw little dots here. These show that we're holding the coil or the pleat in place, and so these bonds are stabilising bonds or hydrogen bonds. So we've still got peptide bonds because we've still got a chain of amino acids, but now we've got hydrogen bonds as well, which are holding them into place. Tertiary level of structure This time we've got more complex folding into a 3D shape. So this level of structure is key to most functional proteins like enzymes. So if we have a look at the types of bonds that are involved here, obviously we've still got peptide because we still need amino acids held in chains. We've already seen how hydrogen bonds can help to stabilise them. We also have what we call disulfide bridges. They form between sulfur atoms in R groups. There are only 2 amino acids that have sulfur groups, that's methionine and cysteine. So if we've got those inside a protein, they can form disulfide bridges. We've also got ionic bonds. They form between charged R groups, so positive to negative. And then the last one is we have hydrophobic interactions. And remember from your membranes work that if a region is hydrophobic, it doesn't like water, so hydrophobic interactions cause parts of proteins to fold into the centre that are hydrophobic. And then lastly, quaternary structure. Haemoglobin is a really good example of a quaternary protein. The reason it's quaternary is twofold. It has more than one polypeptide chain, and haemoglobin's case it's actually got 4, hasn't it? And the other reason that we might classify something as quaternary is if it has a prosthetic group. And remember, prosthetic group is a non-protein group. Now in proteins we only have, if you remember the structure of amino acid, we only have the atoms carbon, hydrogen, oxygen, nitrogen and sometimes sulfur. So if it's not those atoms and it's something else, then we describe it as a prosthetic group. And remember, haemoglobin has an iron, an Fe3+ ion. So that's not part of a normal protein, so we describe it as a prosthetic group. The last thing we need to learn about proteins is we need to learn about the 2 types of proteins. So we'll do a little simple table. Okay, there are globular proteins, haemoglobin's a good example here, and fibrous proteins. So the 2 examples that are on your specification, globular is haemoglobin, fibrous is collagen. Okay, let's have a look how they differ from each other. Well, haemoglobin has 4 polypeptide chains, collagen has 3 polypeptide chains. Haemoglobin has only one type. Haemoglobin has 2 types of polypeptide chains, so 2 types, it's got alpha and beta chains. There's only one type of chain in collagen. Okay. All globular proteins tend to be ball-shaped, whereas fibrous proteins tend to form long, thin fibres. Fibrous proteins like collagen tend to have a structural role. It's obviously structural. Collagen's found in the skin, and that means they're usually insoluble. Globular proteins like haemoglobin often have a metabolic role, such as haemoglobin and it's transporting oxygen, and this means that compared to fibrous proteins, they tend to be soluble. Okay, so that's a quick run through of proteins. So we've looked at the structure of amino acid, how to condense that into a dipeptide, what the bond is called. We've looked at primary, secondary, tertiary and quaternary structure, and we've looked at the difference between globular and fibrous proteins. What you now need to do is you now need to have a look at one of the model answers, have a look at the mark scheme and then try some of the exam questions for yourself.